Okay. All right, we'll begin. I'm happy to welcome all of you to the Myeloid Network's meeting today on June 8th. I want to first remind you that we're having our very first in-person meeting as a pre-conference program at the Society for Immunotherapy of Cancer 2023 annual meeting on targeting myeloid cells in immune oncology, Thursday, November 2nd from 9 to 5:30 in San Diego, California. So I hope you will join us. And Dimitri, Jennifer, and I are the organizers of this. So please contact us if you need more information. All right. So again, this is the Myeloid Network. These are our co-founders. And we are very grateful for support from UC San Diego, AstraZeneca, and Charisma Therapeutics. So today, I'm very pleased that we have a guest speaker, Jelani Zarif, and Jennifer will do the introductions.
Great, thanks, Judy. So I'm delighted to introduce Jelani. Jelani, it's been such a pleasure to get to know you and to learn from you as an fellow early career scientist. Dr. Jelani Zarif is an assistant professor of oncology and the Robert E. Meyerhoff Endowed Professor at the Johns Hopkins University School of Medicine, Sidney Kimmel Comprehensive Cancer Center. The Zarif Laboratory has been studying the molecular mechanisms of immune cells within the tumor microenvironment that ultimately promote tumor growth, therapeutic resistance, and metastasis. The Zarif Laboratory also focuses on discovering and investigating new biomarkers expressed on macrophages that could predict clinical responses to standard of care therapy, specifically in lethal prostate cancer, and I think that's what we're going to learn about today. And he's been the recipient of several prestigious awards, which are including the Prostate Cancer Foundation's Young Investigator Award, the Department of Defense Translational Research Award, and NIGMS T32, and the NCI K22 Transition Award. So very impressive, and we're really very delighted to have you here today. So I'll hand it over to you.
Thank you, Jennifer, for that kind introduction. I'll go to my slides now. Then play from start. I'm assuming everyone can see my slides just fine and hear me just fine.
Everything looks great and can hear you fine.
Great, thank you. So again, thank you to the organizers for having me present our work today at the Myeloid Network, and good morning to everyone. Today, we're going to talk about immune-suppressive TAMs, how we focus on targeting them in lethal prostate cancer, and a little bit about prostate cancer. Since last month's talk was more focused on CAR macrophages, this will look at potentially how macrophages impede growth, therapeutic responses, I should say, in prostate cancer. So prostate cancer, the prostate gland is actually a very small gland. It's the size of a walnut, but it typically causes problems later in life for men, whether it's prostatitis or cancer. And it's actually one of the most common cancers among American men. About 1 in 6 would be diagnosed in their lifetimes. And hopefully, and most cancers are caught at a small focus in the gland. Prostate cancer is also a health disparity among men of direct African descent, and one of the leading causes of death in countries like Martinique and other African nations. So there's a huge field that is studying how genetic differences, et cetera, impact onset and aggressiveness of the disease. Prostate cancer, disparity or not, is considered a cold tumor due to a paucity of immune-suppressive inflammatory immune cells and an abundance of immune-suppressive cell types found in the prostate tumor. There are exceptions for those who respond to anti-PD-1 therapy. Majority of cases in prostate cancer, like other solid tumors, Patients don't respond to checkpoint blockade, but in prostate cancer, there are those that have MS microsatellite instability high tumors. These patients typically have high T-cell infiltrate in their tumor microenvironments relative to those who do not have MSI high tumors. And these patients have been found to respond to checkpoint blockade, but they represent a severe minority of all prostate cancer cases, approximately 2 to 3%. So this demonstrates that immunotherapy of the disease is not a direct barrier to immunotherapy. Prostate cancer tumors have low MHC, ligands that bind to NK cells for NK cell activation, and so poor NK cell infiltrate, et cetera. So these tumors are mostly devoid of anti-tumor immune response cells. Prostate cancer can be treated when it's at a small focus in the gland using androgen deprivation therapy and surgery, radiation, or chemotherapy. Unfortunately, some men who were treated with those modalities early on will relapse to the disease, and the disease will be found outside of the prostate, even if the prostate has been removed. These tumors, unfortunately, do not respond to androgen deprivation therapy and are termed castration-resistant prostate cancer. And castration-resistant prostate cancer goes to the bone, where it makes more bone on top of bone. And these tumors are very hard to treat. There are, Provenge and other modalities that are used to treat them, but it's typically a bit too late. And so this disease is actually fatal. And just some statistics for you who don't work in the field. June 8th, 1993, 30 years ago today, the mortality rate for castration-resistant prostate cancer in the United States was about 33,000 men a year. June 8th, 2023, this year, about the same amount of men will die from prostate cancer. So again, nothing has really changed in the needle when it comes to fatality from metastatic castrate-resistant disease. And so our job is to, in our lab, is focused on this aspect of the disease. So as I said, these are just some clinical studies that looked at metastatic castrate-resistant prostate cancer with anti-PD-1 therapy as a monotherapy. These tumors are really devoid of cytotoxic T cells. In the Topalian paper, as well as other papers that have since come out, there has been no real response to anti-PD-1 therapy for metastatic castrate-resistant disease. There are some ongoing studies looking at prostate cancers that are localized and are hormone sensitive to the therapy, but those studies are now underway. So because of the things that I've told you about, prostate cancers being mostly devoid of cytotoxic T cells, our lab focuses on macrophages, which were actually discovered so long ago by Michnikov, and we're still learning so much about them. So they're laid down during autogeny, and they are resident in multiple organs, and nearly all organs in our bodies, including the bone. And they have different names based on their residence, microglia in the brain, et cetera. I really don't have to explain this to this crowd. They're heterogeneous, and in vitro, in some aspects, they appear to be interconvertible. So in vitro, I forget who may have coined the term M1, M2, I think Mantovani, if I'm not mistaken, in the 1990s. But we've been able, many groups and us have been able to repolarize these macrophages in vitro, and work from Miriam Murad's group has shown that in reality, there are so many different subsets of macrophages in the lung. Her lab studies lung cancer and other cell types within lung cancer, that there are about 15 different subsets of macrophages just in the lung. So the overall ethos and hypothesis of today's talk is really that immune-suppressive myeloid-infiltrating cells, particularly TAMs, promote therapeutic resistance in prostate cancer and metastasis. And so we have some in vitro data that got published a long time ago as a fellow, where we took epithelial prostate cancer cells, co-cultured them with monocytes. These cells end up becoming macrophages that are immune suppressive, and they convert these cells to undergo an epithelial and mesenchymal transition. So when we put these cells into animals, these cells are far more aggressive in vitro. A part of our studies looks at the dormancy of prostate cancer, because clinically prostate cancer can reside in the bone, and those tumors are quiescent, meaning that they have entered like G0, stop dividing, and can come out of G0 and begin dividing again. Very different from senescence. Senescence is when they are beta-gal positive and they will not divide ever again, but they're quiescent. And we believe that either the myeloid cell, the resident macrophages in the bone kind of reawaken these cells out of G0. And then a term that the late great Jeff Pollard would call them was metastasis-associated macrophages, would then home to the bone to promote, or to terminal sites to promote primary and secondary metastasis. And so, so those are the things that our lab studies, and our hypothesis is trying to alter those cells, reprogram those cells so that they can upregulate anti-tumor immune responses. And so today, I'm mostly going to talk about glutamine, but I had to actually go through the mannose receptor to get to the glutamine story. So I'll show you a little bit about the mannose receptor targeting that I started, and we're continuing to study, that led us to the glutamine story. So as a fellow, I worked on macrophages in Kim Pienta and Drew Pardo's, Kim Pienta's lab first, and then Drew Pardo's lab second, and where we came up with a method to get homogeneous populations of macrophages using human PBMCs through leukopaks, and a clinical protocol that we had in the lab where we would use MCSF or GM-CSF for 5 days, and then replace the median stimulants in either cell type. And then for M2s, we would use IL-4, IL-13, and IL-6, and we got really homogeneous populations of M2 in vitro, M2 macrophages. There were multinucleated cells, they were giant cells, 206 high, 163, et cetera, and very phagocytic of mannose, which is some studies that we published. And then we also looked for the M1s. And from there, we wanted to look at glycoproteomic analysis of surface molecules that may be N-glycosylated as a potential biomarker to target on macrophages. And so we worked with Wei Zhang and Weiming Yang, who are in the next building. They have a very, very large mass spectrometry laboratory. And so we took human monocytes right from healthy donors through an IRB protocol, monocytes from either of those donors, in vitro polarized, and differentiated them and polarized them to either M1 or M2, performed SPEG analysis, which looks at glycopeptides that are glycosylated on the surfaces of these cells, then did orbit trap liquid chromatography tandem mass spec, and then used the proteome discovery software. What we found was that there was a differential expression of glycoproteins on the monocytes from, let's say, donor A. And then when we compared monocyte to M1 from that same donor, we saw that there were different glycoproteomic scores or proteins that were N-glycosylated. And then we compared that to monocytes to M2s and M1s to M2s. And so we wanted to see what these large differences were. This is the fold change in glycoproteomic analysis, showing that these cells have all have different glycoproteomes, even if they're from the same donor. And so one of the top hits that we found was that the mannose receptor, which is probably no surprise to anyone on this call, was heavily N-glycosylated. Now this is for those who have forgotten their mass spec and biochem and haven't done the NMR in a while, this is a mass spec plot. And this is a peptide sequence showing that asparagine residue is heavily glycosylated, shown in red. And these are the different peaks with the mass to charge ratio on the x-axis. And when we looked for PBMCs are the monocytes from donor A and donor A, and M1s from donor A and donor A from M2s. You can see that there's a high intensity of 206 expression on the surface, and this is at a 1% false discovery rate. We confirmed this by Western blots and QRT-PCR. And so from there, we wanted to look at 206 expression in prostate cancers. So we worked with our clinical pathology team here, Angelo DiMarzo and Tamara Lotan, who constructed prostate cancer tumor microarrays. So these are small bits of different tumors on a very big slide. We worked up the antibody for 206. And when we looked at, normal prostatic tissue and distant metastasis, you can see a substantial difference in 206 expression by macrophages. Prostate cancer has something called a Gleason score. That is, it's scored when it's localized disease. So 3+4 is a clinical pathology diagnosis. So this is localized prostate cancer, and indicative of localized disease. You lose basal cells, integrins change, and there's a fusion of these glands which secrete prostate-specific antigen. You see that here, but you also see an increased 206 stain compared to normal prostatic tissue. And we looked at many different lymph node mets. We saw 206 expression high. And so we quantitated a lot of these by using the mass area ratio. And what we found was that castrate-resistant disease had the highest infiltrate of 206 positive macrophages. We also looked by IHC at dual markers, so 206 and 68, looking in aggressive prostate cancers. This is just one of many that we published, where we were able to succinctly see that CD68 and 206 cells were high in the tumor microenvironments of prostate cancer, and they were on the same cell. We also have a rapid autopsy series here where we looked for macrophages in patients who consented to have tissues studied at the time of their demise. These men died of castrate-resistant prostate cancer, unfortunately. And we had a series of about 30 cases that we did fresh that day. And so we did 45 positive cell enrichment and then stained for 163 and 206, the scavenger and Mannose receptors respectively. And you can see just these are two different patients where 206 and 163 positive cells were high among the CD45 positive infiltrate. When we tried to look for cells that were high in CD86, we saw a paucity of these cells. This is a liver met. We saw similar high expression of 163, 206 dual positive macrophages, but a paucity of macrophages that have CD86 expression. In prostate cancer, I showed you that technetium-99 bone scan earlier. That was from one of Kim Pienta's figures and papers from a clinical study that he did. And so we looked at bone mets. So some of these bone mets were really too small or too hard to try to get a single cell digestion. So we performed immunohistochemistry using the same antibody that we use for the TMAs. And we were able to see that 206 positive infiltrate in bone metastasis of prostate cancer. And so by happenstance, I was at NCI and I ran into a colleague named Clayton Yates, who was designing a peptide that bound to the Mannose receptor. And so, so this Mannose receptor has a fibronectin type 2 domain, and he designed these ancient peptides made by bacteria to fit into that fibronectin type 2 domain and cause a structural shift and change in the Mannose receptor where it's endocytosed and causes increased caspase-3 activation. And so they were working on it with Udo Rudloff, who's also at NCI, and pancreatic cancer models. And so we were working on the human macrophage story. And so we began testing some of these peptides in our human macrophage models. And so what we were able to find in collaboration with Clayton Yates was that this peptide RP182 was able to bind the mannose receptor, be endocytosed within 2 hours. And within that 2-hour window, the TAMs were either reprogrammed or they died. And so the cells had kind of two fates. And there's a lot of data in the paper. I'm only showing what we helped contribute to that story that was published in 2020. But there was increased caspase-3 and caspase-7 activation. And when we tested our model, M1, M2 for in vitro testing with like an MTT assay, we were able to see that the RP182 was able to kill the macrophages. This was some other studies looking at CD86 and 206 expression after a period of time, and we saw that there was a drastic change in 206 expression after RP182 bound to these macrophages within 30 minutes. And then we looked at cytokine secretion, either by multiplex cytokine analysis, QRT-PCR, and the like. And we were able to see that these cells, CD11B F480 macrophages isolated from tumors, were able to increase cytokine secretion in the cytokines listed. Since then, a new iteration of this peptide, RP832C, is being tested, and we're working on this in collaboration with Clayton Yates, who's now, his office is next door to mine. He was at Tuskegee when those studies were first published, where 832 is able to bind to the mannose receptor for a lot longer than RP182 and reduce prostate cancer tumor growth in terminal sites relative to control. And this is showing docetaxel, which is DTX. And so while we were working on the mannose receptor story, there was a really nice paper that came out from Agios. And shout out to anybody who worked at Agios who's on the call. What they did was they looked at mirroring monocytes and deprived them of glutamine and looked at polarization states. And they showed really nicely that glutamine deprivation altered their polarization states. And I was like, wow, I'm working on these human monocytes. Maybe it works in my model too. And again, thank you to those who worked at Agios who might be on this call. And so I basically repeated the experiment that they did it was an immunity paper. My paper wasn't in immunity, unfortunately, but still got it published. And so what we did was we took those human monocytes that we were differentiating into DCs and M1s and M2s and just took glutamine away. And what we were able to find just by flow is that CD86 shown in orange here, these are the glutamine-treated, glutamine-deprived samples. The CD86 increase, there was a 206 decrease, 163, IL-4 receptor alpha decrease, as well as the ECAD-herin. Really, we saw some phenotypic change in the macrophages, but it led to newer ideas. And so around the same time, a colleague upstairs, Jonathan Powell, was working on targeting glutamine, glutaminase with lots of different methods. And so they wanted to study how glutamine affected T-cell proliferation, et cetera. And so this is from Min Hee's paper. She was a graduate student with Jonathan Powell, where they looked at kinase effects on T cells, MDSCs, and TAMs. And so with using a glutamine antagonist that they developed, they were able to show that TAMs were able to increase their MHC peptide production in recruiting T cells and increase tryptophan secretion for T cell proliferation, which promoted killing of tumor cells. This was I think this paper was published in Science, this was the paper after the Science paper. So the Science paper was Bob Leone's paper, and then this one came right after. And so, the prodrug that Jonathan Powell and Barb Slusher designed is called JHU083. It's named after where it was created, okay? So I had no say in the name. And it's a name I can actually pronounce. Some of the drugs out, the inhibitors, I was like, who came up with the name? So this is something I can actually pronounce, okay? So it looks like glutamine, Don does. Don was very toxic. It caused gastrointestinal issues in the 50s and 60s. So it's no longer used. And for those who did okay in organic chemistry, you can see that glutamine looks a lot like Don, okay? And so does JHU083, but JHU083 is a prodrug where this leucine linker and ethyl group get cleaved once it gets inside of a tumor microenvironment and becomes Don, okay? So Barb Slusher and Jonathan Powell are here. They are the ones that created this drug, and this drug is now, I think, marketed through Drayson, which is a pharmaceutical company that grew out of this collaboration. They tested this drug in several other studies that I'm not involved in. They looked at pigs metabolize glutamine the same way humans do, so they showed that there was no toxicity in pigs. They also showed it preferentially delivers the prodrug to tumor cells in the plasma compared to Don. And the prodrug does not release in gastrointestinal tissues, which is a huge, huge hurdle that this drug could have not overcome because it's a Don prodrug. So we became very interested in glutamine targeting, again, with the Agios paper and with other things that we were doing in our lab. And so I decided to write a K award on utilizing or targeting glutamine. And we had JHU083 here. And so we were going to first try to get a lot of bone met samples to look at glutamine signatures and bone metastasis. But luckily, the Kafori et al. Paper came out before we even got started. And so they did a lot of the experiments we wanted to do. So they dissociated bone. They had tumor-involved bone, bone-involved marrow, distal marrow, and then they had normal bone from like fractures. And so they did single-cell RNA sequencing on all of these and found some incredible things. And so what we then did is we just mined their data, and a graduate student lab who's an expert at that, Alex Lee, helped us with this. And so we were interested really in glutaminase and glutamine synthetase. And so we looked at tumor-associated macrophages in benign, distal, involved marrow, and tumor. And you can see that this is glutamine synthetase, GLUL, that TAMs have a really high level of glutamine synthetase. We also looked at glutaminase in tumors, and you can see that it's a high expression by single cell. And so to really study prostate cancer around the time of the first JHU083 paper from the Powell group, we had published a syngeneic autochthonous MYC-driven prostate cancer mouse model, because in prostate cancer, we typically have very limited models that mimic disease that you see in humans. And so what we did is we back-crossed the Charles Sawyer's high MYC model, which has the androgen receptor and MYC, which are two of the major genes that drive prostate cancer metastasis and progression. And they're also luminal cells, so these are cytokeratin 8. We back-crossed this model, which is on the FVB model. We back-crossed it to C57 Black 6. And then these tumors formed within the lung and within the prostate, and we consistently isolated these tumors and grown them in vivo only. They do not grow in vitro. And they have a lot of the characteristics of human disease. They're synaptophysin negative. This is a pancreas islet. Synaptophysin is of a neuroendocrine or a small tumor type, which is a weird thing that's sometimes seen in aggressive disease, but does not mimic adenocarcinoma. These tumors also maintain their androgen receptor expression during metastasis, and when they metastasize, they form new mets on top of bone. So this is the skull, this is the femur, this is gross pathology of the femur. You can see the bone met on top of the femur there. And these localized tumors, as well as the aggressive ones, are infiltrated by tumor-associated macrophages that have a paucity of T cells. Finally, we've also investigated this model, whether it responds to anti-PD-1, and after about 100 days of treatments, we find that it does not respond to anti-PD-1, which is similar to what we see in patients. And so this work was done and led by Manali Praharaj, who graduated 2 weeks ago from Johns Hopkins School of Medicine in the pathobiology program, and is now at Takeda. And she worked on JHU083 and looking at really urologic tumors, so bladder cancer, prostate cancer. So the two models, three models here I'm showing are B6CaP, which is the model I just showed on the previous slide, the histology. JHU083 is able to substantially reduce tumor growth in this model. There's another cell line-based model called RM1 that we utilize. It's really aggressive. You can get really big tumors after just 20 days, and that's with 50,000 cells implanted, okay? And so JHU083 treatment reduces tumor growth in this model. And then MB49 is a bladder cancer model that's really aggressive too. And these tumors are urological, and they are highly infiltrated with tumor-associated macrophages based on lots and lots of clinical studies. And we can see JHU083 is able to reduce tumor growth in vivo here. And that's not the end of, really the end of the story. We began looking at the anti-tumor immunity and anti-tumor immune responses after JHU083 treatment, and whether it was partially dependent on T cells. And so what Manali and others in the lab would do is at day 3, give either anti-CD4 or CD8 into the mouse, and then implant the mouse with tumors, and then give additional doses of JHU083, and then perform necropsies to look at dependence on T cells. Okay, so this is a kind of a busy slide. We also looked at survival analysis, but JHU083 was substantially by itself able to reduce tumor growth. Anti-CD8, so this is the bioexo antibody with JHU083. Tumors are smaller. Anti-CD8 alone, tumors are even bigger than the isotype controls. And we also looked at, this is in B6, that was in MB49 and B6CaP. We also saw a reduction, just JHU083 alone, in mice that were treated with just JHU083 alone, or with anti-CD8 had increased survival relative to the control. We also did anti-CD4. This is just a bioexo antibody. And we see some, again, partial dependence on T cells. And so that really warranted us to think a little bit more about the functions of reprogramming these myeloid cells, particularly macrophages that are really abundant in these tumor types. And so one experiment we did is the adoptive transfer. And so we would either take MB49s, implant them into a mouse, give JHU083, and then harvest tumors, isolate the macrophages from these MB49 tumors using CD11B, F480, lyse, and CD3 negative, CD45, all those gates that you probably typically use with your flow work. Mix those macrophages with MB49s, and then implant a new mouse, and then look at tumor growth after that. We also did this with infiltrated monocytes. So we see this in tumors. The thought is that monocytes traffic to the tumors, differentiate in the tumor once they see specific signals, and become tumor-associated macrophages. The other thought is that if it's localized prostate cancer, the macrophages end up becoming the resident macrophages become immune suppressive and tumor associated macrophages if they aren't already. And so we were able to isolate monocytes, mix them 1-to-1 with MB49s, and also measure growth after that. And so what we see, and this is with MB49, so these are TAMs, the adoptive transfer, untreated macrophages with MB49. You can see that these tumors grow nice. MB49s alone, these tumors grow really nice, but macrophages that were treated with JHU083 and then mixed 1-to-1 and then put into a new mouse, after 27 days, we can see that these tumors were substantially smaller. We also did an additional dose of JHU083, and it was not significant. It was not statistically significant compared to just the JHU083 treated macrophages, just treated one time. We also looked at immune-infiltrating monocytes, or tumor-infiltrating monocytes, and we see the same trend that JHU083 treatment of monocytes and mix 1-to-1 with MB49s and put into a new mouse, they are able to, again, prevent or cause tumor growth inhibition in vivo. We then began looking at transcriptional changes in the B6CaP tumors. We also looked at MB49s, but for the sake of time, I can't show all the data. So we did two different approaches here. So we did an early treatment approach. So we let tumors grow, and then on day 25, we treated with JHU083 and then isolated those tumors 7 days after treatment. And then we did some late treatment. So we did initial dose, and then a later dose, and then we isolated and performed bulk RNA sequencing on tumor-associated macrophages from these B6CaP tumors. And so what we found, and this is probably a presentation in and of itself, is that JHU083 shifted the density of subcultures, or clusters, I should say, of TAMs at both time points. So here's our control. And we have, there's a manuscript that looks at all the different types of macrophages. So TAM 1 through 5, inflammatory, proliferative, glycolytic, et cetera. And we saw shifts. You can even see this population shift of glycolytic TAMs shifted here. And then we looked at DEG scores that went up and down within the, after the late time point using single cell RNA sequencing. We also, within those, we also found that JHU083 promoted TNF secretion and proliferation in tumor-associated macrophages isolated from these tumors. So this is just showing that data here, that there's increased TNF. Even in macrophages that are 206 positive, TNF was secreted, indicative that it's not always just a surface marker, it's the function of the macrophage. We looked at MHC class 2, CD86 positive TAMs, and we can see that JHU083 increased TNF in them, as well as the monocytes. These cells were more proliferative. I remember Judy Varner saying this on the last Myeloid Network meeting, is that that some macrophages are able to proliferate. We definitely see that with Ki67 staining, especially of those that are 206 positive. And this gave us some pause a bit because, you know, I showed you all the data earlier about 206 and targeting 206 using this peptide, but the function of the cell has changed, even though they still express 206. And then JHU promoted some inflammatory signaling and proliferation in TAMs. This is just more data looking at that. So we looked at the mitotic spindle. These are DEG scores that are upregulated, TNF, G2M, inflammatory response. These are all up. We looked at the proliferating TAM subcluster, and you can see that these are up as well. We looked at late, so we have TNF alpha is also up, as well as the inflammatory response. And G2 checkpoint mitotic spindle. So we then wanted to ask JHU083, which is able to become a prodrug in the tumor microenvironments of MB49 and B6CaP tumors, which are highly abundant in tumor-associated macrophages. We are seeing that these cells are more proliferative, and we see an increase in TNF, and this TNF is being secreted. So what effect is it having on tumor-associated macrophages, as well as their function? So with help from Elizabeth Thompson, who was a postdoc with Jonathan Powell, looking at metabolism, we began looking at GLUT1. So just to walk through this a bit, glucose binds to GLUT1 and becomes becomes pyruvate, and then can go shun out as lactate or go into the Krebs cycle as pyruvate. And here's our little Krebs cycle here. And this happens in the mitochondria, as everyone knows. And so we wanted to look at GLUT1 changes after JHU083 treatment of macrophages isolated from either B6CaP or the MB49. And what we can find is that hexokinase 2, which is HK2, as well as GLUT1, these cells are increased, as well as the percent of hexokinase 2 M1 macrophages are increased, as well as M2s. We also looked at TORC signaling. We have a whole lot of data on that, but I couldn't fit it in. I'm actually kind of tight on time as it is. But we looked at TORC signaling as well as glycolysis, which I'll talk a little bit about later. We saw that JHU083 induces parallel metabolic changes, and we worked with Liang Zhao, who was also in the Powell lab, and particularly in purine metabolism, looking at a protein called FGAR, which is formyl-glycine amide ribotide. It's an intermediate for purine nucleotide synthesis. And so we isolated tumors and digested them, sorted TAMs out, and then looked at metabolic metabolites from these TAMs using liquid chromatography tandem mass spec. We found a litany of things. As you can see, JHU, we had 3 different samples, lots of genes turned off relative to metabolic function of FGAR. And then the 2 proteins that we found that were the highest and most differential compared to the controls were guanosine and FGAR, which again is an intermediate for purine nucleotide synthesis. And so we wanted to see how JHU083 was altering glucose metabolism. And so we began to do some classic experiments using heavy glucose or heavy carbon to do a glucose tracer. So we would inject the heavy glucose into the tail of mice that had tumors, perform rapid digestion, sort those cells, and then do polar metabolite extraction, and then look, based on carbon tracing, look at where the directionality of glucose. And so TAMs typically are glutaminolic, so they love glutamine, but the inflammatory macrophages, and this is work of many others, and some of your mentors who are on the call or might not be on the call, are actually glycolytic. And so we wanted to see if JHU083 could induce a broken TCA cycle. And what we found is that it does, and it increases IL-1 beta production. And so particularly, we looked at lactate, we looked at everything in the TCA cycle. We didn't see many changes in the labeled fraction. But then when we looked at the abundance of alpha-ketoglutarate, we saw that there's a decrease in alpha-ketoglutarate as well as succinate. That is, just to remind you of the TCA cycle, it decreased the succinate because this TCA cycle is broken here due to a decrease in alpha-ketoglutarate. Sorry about that. And then we looked at the other features of the Krebs cycle after JHU083 treatment. And what we found again were a broken TCA cycle and an increase in IL-1 beta. So far I've shown you that JHU083 is able to reprogram tumor-associated macrophages by increasing IL-1 beta, TNF secretion and proliferation, as well as, I didn't get the chance to show all the TORC data that we have, but definitely GLUT1 through hexokinase 2. And so what is the effect on the function of these TAMs? And so we wanted to really look at, see if these cells were reprogrammed to a point where they phagocytosed tumors. It's one of the original functions of macrophages discovered by Michnikov, right? And so we did some experiments looking at this, where we labeled MB49 cells with RFP, and then treated those cells, those tumors in vivo with JHU083. And then we looked at the non-treated versus MB49 only. And as you can see, the MB49 RFP JHU083 treated tumors are much smaller, as shown here. And then we began looking at MB49 cells only that were RFP, that weren't RFP labeled. And then we looked at RFP labeled vehicle-treated MB49s. And then we looked at those that were treated with JHU083 to see if they'd been phagocytosing tumors. And so what we were able to find is that these macrophages had a higher level of RFP, don't have any RFP off the top, but now they have RFP because they're phagocytosing the MB49 cells after JHU083 treatment. Another look at this was we would perform in vitro experiments using DON, which is the non-prodrug form of JHU083, where we would label macrophages with PKH26 and PC3 cells with CFSE, mix them together, and look under the microscope or do flow cytometry. And so that's what's shown here. And so these are the PC3 cells. These are a classic prostate cancer cell used in just about every prostate lab I've ever been in. And then macrophages that are PKH26. And you can see red and green mixed together. If you're red and green colorblind, no problem, I got a fix here for you. Because we did flow cytometry using these cells, and you can see after we add glutamine or glutamine is present or we take glutamine away, you can see these macrophages have an increased phagocytosis of these PC3 cells. If we use DON for 9 days, you can see that that jumps from 2% to 21.8% of phagocytosed cells, and these are the histograms here. JHU-83 also has an effect on the tumor. And so we looked at GLS1 and GLS2. So these are glutaminase 1 and 2, and we can see that those proteins are reduced. We looked at glutamine synthetase, GLUT1, and beta actins are loading control. And that's, we also did this by flow, so not really a substantial change in GLUT1, but definitely a reduction in GLS1 and 2. We surprisingly found mannose was upregulated as a after JHU083 treatment, so was phosphatase, pyrophosphatase, when we did proteomic analysis. And we believe that this might be one of the reasons why 206 stays expressed on these macrophages, even though they're making TNF and doing all these other things that might be associated with an M1-like macrophage, so to speak. We also looked at the tumor cell itself. And so we looked at MB49 and B6CaP after JHU083 treatment because tumors also are highly dependent on glutamine. It's in just about every cell culture media, especially Eagle's minimum essential media. And so we saw that glutamine went up, actually glutamate went up, and as well as FGAR in both of the cell types after JHU083 treatment, which again is associated with purine nucleotide synthesis. Prostate cancers and other cancers are highly dependent on MYC. We saw a decreased phosphorylation of MYC at these specific amino acid phosphorylation residues, as well as HIF1. And this was done by Fan Shin, who's the second author on this manuscript. We also looked at cell viability and apoptosis of these tumor cells after JHU083 treatment, which again, reprograms the TAMs, but the tumors are part of their microenvironment too, and they are also partially affected. And so this is really a summary cartoon of everything I've shown this morning, where tumors, urologic tumors that we've tested after JHU083 treatment, there's an increase in IL-1 beta in tumor-associated macrophages, TNF. These cells are more proliferative. They maintain their 206 expression, but mannose is also in the microenvironment as it's part of the glutamine synthesis pathway. And I didn't get a chance to show the MYO1E, but this is a protein that's affiliated with phagocytosis. And I did show the phagocytosis data, but this is a MYO1E dependent mechanism, as well as these cells are secreting more TNF relative to the non-treated controls. After we deplete CD8s, we saw that there was some dependence on T cells, but it appeared that the macrophages were doing a lot of the heavy lifting and phagocytosing and upregulating anti-tumor immune responses to recruit these cells to the microenvironment. I didn't have a chance to show that after JHU083 treatment in single-cell RNA sequencing, we see stem-like T cells in the tumor microenvironment, as well as a robust increase in NK cell trafficking. That was like 5 or 6 slides of data, so I didn't have a chance to show that. And that this pathway of glutamine and glucose, it regulates pyrimidine levels, purine synthesis through FGAR, glutaminolysis through GLS1, and MYC and HIF1 alpha are altered in the tumor cells that we looked at, which are bladder and prostate. Those are the tumor types that we study. So this is the conclusions of everything I've shown today. The first story was about 206 and RP182, another peptide that binds, that is able to specifically target TAMs. We're continuing to work on this in our syngeneic model and combine it with docetaxel and other agents that aren't, that prostate cancers become resistant to over time. The majority of the work that I've shown today was on JHU083, which is a glutamine prodrug developed by Jonathan Powell and Barbara Slusher. The JHU083 caused tumor growth inhibition in vivo in every model that we tested, and it induced a tumor cell phagocytosis, T cell infiltrate, and NK cell infiltrate. These cells were stem-like. JHU083 treatment induced a broken TCA cycle in tumor-associated macrophages isolated from these microenvironments that are mostly glutaminolic. They're now glycolytic, and they increase, you had an increase in IL-1 beta. These cells are more proliferative as well. And JHU083 altered metabolites within the tumor, specifically that of mannose. So this work was led by Manali Praharaj, shown here. She's now at Takeda. She's a Johns Hopkins grad, as well as Fan Shin, shown here, a postdoc in the group. Tom Nerschel, Xiao Shu, and Alex are here, and Kenneth also worked on the paper. as well, and it's under review. We had a lot of help from colleagues here, Jonathan Powell, Barb Slusher, who developed JHU083, helped us with ideas. Edward Pierce and his group in Insights, Liang Zhao, Dr. Pardo, our clinical and pathology colleagues for helping us with the acquired tissue, et cetera, for the TMAs and other work that we want to do to really see if our targets are actually useful in clinical disease. This work was funded by my K22, Allegheny Health Network, Prostate Cancer Foundation, startup funds from the Bloomberg-Kimmel Institute here. And I thank you again for giving me an opportunity to talk about our work. I'm happy to take any questions. Thank you.